Welcome to In Your Neighborhood. I'm your host, Chris O'Rourke, as we continue our series, Conversations on COVID-19, except this time we have a slightly different format. In the past, we've done question and answers over Zoom. Now we have a Zoom presentation for you, made recently by Dr. Tammy Tobin from Susquehanna University. She talks about the facts and fiction behind COVID-19.
Hello, everybody. My name is Tammy Tobin. I'm the chair of the biology department at Susquehanna University, and I would like to thank all of you, our alumni, faculty, staff, students, and prospective students, for attending my class today, Understanding COVID-19, Separating Fact from Fiction. As we said in our introduction to this session, my goal for today is to help give all of you the scientific knowledge and access to reasonable resources that will help you all make decisions for your own behavior regarding COVID-19 that are based more in science and less in social media. To that effect, we're going to talk about the biology of the virus. We'll talk about how COVID-19 spreads. We'll talk about how we as individuals can slow that spread. I'll tell you a little bit about where we are with testing, treatment, and vaccination, and then leave you with some resources that you can use moving forward. So COVID-19 is caused by a virus called Severe Acute Respiratory Syndrome Coronavirus 2, or SARS-CoV-2. The virus is shown right here in the middle. You can see that it's a deceptively simple-looking little creature. It consists of RNA, which is its genetic material, an envelope, which is the gray substance surrounding it, that comes from our own cells, and then proteins. And the protein that I really want you to focus on here are those red glob spiky things on the outside of the virus. We're going to be talking about those a lot today. Those are referred to as the spike glycoproteins, and they're what the virus uses to bind to our cells so that it can infect us. So keep those in mind as we move further on into this class. Now, fortunately for our situation with COVID-19, coronaviruses are well studied. In fact, we know that there are hundreds of different coronaviruses that infect all different kinds of animals. Of those, only 7 are known to sicken humans, and most of them cause mild disease. In fact, 2 of the most common causative agents of the common cold are coronaviruses. 3 coronaviruses are known to cause severe human disease. The first is SARS-CoV, that causes SARS. The next is MERS-CoV, that causes Middle East Respiratory Syndrome, or MERS. And the final one is SARS-CoV-2, that we now know causes COVID-19. One of the first areas that I want to talk about, separating fact from fiction, is hitting a lot of the news media and also a lot of social media now, and that is, where did SARS-CoV-2 come from? In order to answer that question, we really need to go and to understand how we know where this virus came from. I want to take you back into the original SARS and MERS stories. So in order to figure out where a virus comes from, what we do is we sequence that virus's genetic material. In this case, coronaviruses have RNA. We sequence that genetic material and we compare the sequence of our virus to the sequences of all of the coronaviruses that infect animals. It can be even worldwide. We look for genetic similarities with the idea that the 2 sequences that match the most closely will be the most closely related. This is just like we do in human DNA sequencing. My DNA sequence is going to be much more similar to my brother's DNA sequences than to anybody else's sequence out there in the audience, because we're related to one another. We share parents. So we sequence these genomes of the viruses, we compare them to one another, and then we assemble something called a phylogenetic tree, which is very similar to a family tree in humans. And so let's see if my little arrow can go down there. It can. Right here at the base, if you can see my arrow at the very base of this tree, would be where the common ancestor is for all of the types of viruses above it. And again, we know that because we can tell from sequence differences that all the sequences derived from that one. You can see that that one, if we follow it up, is a bat sequence. And this is the study that was done for the SARS virus, the original SARS virus that came out in the early 2000s. So that ancestral one was definitely a bat one. And so when we compared humans to bats, we saw a lot of similarities. What was interesting, though, was that the most closely related sequence to us was actually found in something called the civet cat that's also in China. And I should say the bat was the horseshoe bat. You can see that here. This also lives in China. What became apparent then by looking at the sequences was that this virus originated in bats. It then moved into the civet cat, where a few changes happened that we could follow, and then it moved from the civet cat into humans themselves. So again, it originated in bats, moved into civet cats, and then into humans. When we looked at the MERS outbreak in the Middle East, we found, not surprisingly, that also originated in bats, although I didn't know until that time that bats lived in the Middle East, but they do, and that the intermediate host, the one that it jumped to from bats before infecting humans, was actually camels. So scientists are now using exactly that same sort of analysis to determine where SARS-CoV-2 originated, and not surprisingly to anybody, we found that it also originated in this horseshoe bat that lives near the Wuhan area of China. In fact, the human virus and this bat virus have about 90% identity, so they're very, very similar. There's a little bit of a problem with that particular lineage, however, and that's if you think back to that red spiky protein that I told you about at the beginning of the talk, where that virus binds to the host cells. When we look at the sequences necessary, the protein parts necessary to bind to our cells, they don't match this bat at all well. In fact, what they match is the protein sequence from the pangolin, this scaly anteater that also lives in China. So it seems to have this pangolin binding site, and the rest looks like bat. And I just want to show you a little bit more about what this looks like. So if we look at this figure here, if we go down to the very bottom where I've got this circle, I'm just circling a tiny little bit of this virus protein. And then down here below where we've got these colored boxes all lined up, the top row shows the amino acid sequence, the protein sequence of that short part of the spike for humans at the top, bats in the middle, and pangolins at the bottom. And this is specifically the section that binds to the receptor on human cells. Any place that's really important for binding is in a box. So you can see in the human sequence, there are 6 different boxes spread along there. When there's identical sequences between humans, bats, and pangolins, you can see that the boxes, you will have all the same color and the boxes will be joined. So in this very first amino acid to the farthest left, human, bats, and pangolins are all identical. However, when we move to all of the other amino acids, the bat isn't the same at all, but the pangolin is. So it looks like the binding portion of our coronavirus either came from a pangolin or some other animal that has a similar sequence. The reason that we don't think the pangolin is where it came from originally is because the rest of the virus doesn't match our virus at all well. So again, what we know is that this virus originated in bats and that it either went to humans through pangolins, where maybe the bat and the pangolin sequences recombined, or through another intermediate species that matches us much better, and then onto humans. It could also have gone straight from bats to humans and just accumulated all these differences one at a time until it finally became the kind of efficient killing machine that it is now. So it started with bats, and then it changed so that it could infect humans and cause disease. And human disease all starts pretty much in the same place, and that's right up here in your nose. So the virus gets into your nose. We'll talk about how it does that in human-to-human transmission in a little while, but it ends up in your nose, and all viruses have to bind to a cell receptor on your cells in order to infect you. And for this SARS-CoV-2, the receptor is called the ACE2 receptor. The virus is able to infect humans specifically and some other animals, but as I show down here, not pig noses, because we actually don't share enough sequence similarity in our ACE2 receptors for the virus to infect them. We can also be infected. You can see just a little bit of an eye up here. If we have contaminated hands and rub our eyes, the virus can bind to cells there. Once it binds to cells, the rest of this diagram really just shows you how the virus goes from one virus to lots and lots of progeny viruses. So the virus needs to get into the cell, so it shows it going down in this little part here that says fusion. It gets into the cell, and then it releases its genetic material, its RNA, into the cell. That RNA then serves two purposes. It serves as a template to make all of the virus proteins that need to be made to make progeny viruses, and it serves as a template to make all of the virus chromosomes that are necessary to make progeny viruses. So that's really what's being shown here. Using our cell's own ribosomes and other cellular compartments, it makes all the virus proteins, it makes all the virus chromosomes, it assembles them together. You can see it going up here again to the upper right. Those virus progeny assemble, and then they go out of the cell. While they do that, they're not doing the cells any favor at all. They've basically converted the cell from doing whatever it's doing in your nose to making viruses, and that oftentimes, very often, kills the cell. So now we've gotten that virus from bats into humans, and we've got a human that's taken just a few viruses and now made millions of viruses that they are now exhaling. When those viruses are released then, or when those viruses are released, they can either go out into the environment again, or they can go and contact other cells. And the way that COVID spreads to other cells really helps us understand how it causes those symptoms. So first of all, SARS-CoV-2 can infect and kill a wide variety of cells, from our cells in our noses, to cells in our lungs, to kidney cells, cells in our blood vessels. It can infect our gastrointestinal cells, and that is why some people get nausea and diarrhea when they get COVID-19. So it really causes a lot of symptoms because it's infecting lots of different kinds of cells. Now, most of the symptoms that are caused by COVID-19 appear to be done by our own immune response to that virus infection. So to understand that, and in specific, it's something called an inflammatory response. So if you think about it, when you get an infection in your finger swells up, it gets red, it gets hot, it gets swollen. That's an inflammatory response. And one of the things that's happening is fluids and cells from your blood vessels are flooding into that infected area so that they can combat that infection. They can combat the bacterium or whatever else is in there and get rid of it. So an inflammatory response is a very normal part of our immune response. However, when an inflammatory response happens in our lungs, it can be very dangerous. So I'm looking over here now to our little picture of the lungs, and you can see the lung here, and blood vessels and air vessels, and down at the very ends of all of our air vessels are little sacs called alveoli. And you can see down here in the bottom, this is an alveolus. These are teeny little sacs, and when you inhale, oxygen goes into these alveoli, and it goes across this very thin layer of cells from the alveolus into your bloodstream. It's picked up by red blood cells, carried to your heart, and oxygenated blood is pumped throughout your body so that your cells and organs can work. That blood then goes back to the alveoli and releases carbon dioxide. So when you inhale oxygen. When you exhale carbon dioxide, and that all requires that the alveoli be healthy and not filled with anything other than air. The problem is that when an inflammatory reaction happens in your lungs, and that's shown to the right here, all those fluids and cells that we talked about going into your finger when it was getting inflamed actually go into the alveoli. And so now these sacs are filled with fluids and with cells, and you can't get any oxygen in. So that's why people end up on ventilators when they have very severe COVID-19 symptoms, because they simply can't get enough oxygen into their lungs when this inflammatory response is happening. Another somewhat confounding symptom with COVID-19 is that some people seem to be doing fine very early in an infection and then all of a sudden get very, very ill. And it's thought that this might be due to something called a cytokine storm. Cytokines are small molecules that signal our immune response and actually are very responsible for signaling an inflammatory response. And it's thought that so a cytokine storm really is an unregulated release of lots of cytokines that causes an unregulated inflammatory response. It's thought that what may be happening is that in some individuals, they feel fine very early in the infection because their immune system is not responding very strongly to the virus at first. So the virus ends up moving from their noses to their lungs, throughout their bodies, and by the time the immune system notices it, it's everywhere. The virus is everywhere. And that triggers the immune system to just overreact and say, oh my gosh, there's a virus everywhere, we've got to kill it. And it just floods the system with cytokines, and the inflammatory response can go systemic. And so you move very quickly into this situation where the alveoli are flooded. By the way, that's pneumonia. And you can also get clotting of blood vessels and organ failure and death. So cytokine storms may be an important part of this pathology. Some of the other things that we still don't understand very well about COVID-19 symptoms are we know that some folks get sicker than others. Age seems to play a pretty major role. Most of the people who get but not all of the people who get very sick and die are older. So age may play a role. That may be because of the nature of our immune systems as we age. There may be other underlying factors. Biological sex seems to play a role. So evidence suggests that men have a harder time clearing the virus and that they may also be more at risk for adverse effects than women. Again, we're not sure if that's because of biological differences, because of lifestyle differences, or both. And of course, we know that underlying conditions, diabetes, obesity, asthma, other lung conditions, can predispose people to very serious complications. Okay, so I'm going to back up again. We started in a bat. We moved up a human's nose. That virus spread all around the human's nose and their lungs, and now that human is getting ready to exhale and spread that virus to the next human. So the next thing is, how does SARS-CoV-2 spread between humans? This is something we do understand very well, actually. SARS-CoV-2 is spread between humans in three major ways. The first is shown in this picture. Everybody, when they exhale, sort of spews out water droplets. And in a SARS-CoV-2 infected individual, those water droplets can have live virus in them. So they go shooting out in their water droplets and head towards this next person. Now, it's pretty clear, we're fortunate, SARS-CoV-2 droplets mostly fall to the ground within about 6 feet of the infected person. And I say mostly because there are some people that spread water more effectively than others, and certainly if you're coughing or sneezing, those things are going farther. But with normal breathing and talking, they fall to the ground more or less in 6 feet. So when we start talking about how we can prevent the spread, this is one of the reasons why we say everybody needs to be at least 6 feet from each other, preferably even farther would be great. We also know that close contact spreads SARS-CoV-2 very effectively. So close contact with an infected person, kissing them, holding hands with somebody, and then rubbing your eyes, things like that. So close contact can spread SARS-CoV-2. And also these droplets, when we exhale them, fall down and hit things around us. So they hit our cell phones. They would hit my computer in front of me and the table in front of me. When my hands touch those items, my hands would get contaminated. And then the next time I touch a doorknob or the handle to a grocery cart or anything like that, I could be spreading that virus from me to other people. Likewise, if somebody else has spread their virus to that contaminated surface, if I touch that, I could also become contaminated. There have been studies that indicate that SARS-CoV-2 will live anywhere from about 2 hours to a few days on most surfaces. That depends on the nature of the surface and the temperature. So colder temperatures and harder surfaces the virus survives in an infectious state longer than some other types of surfaces. So we'll also talk about disinfecting things as a way to prevent the spread. All right, so bat up a person's nose, out of that person to another person. And that takes us to the next big topic, is how did that SARS-CoV-2 pandemic spread globally? We know the answers to this very much the same way we know where SARS-CoV-2 started, because what we do, or what scientists have done, is they've sequenced the SARS-CoV-2 genome in infected individuals all over the world, and they've compared those sequences to each other. And as SARS spreads from one part of the world to the other, and all of the evidence indicates right now that it did start in the Wuhan area of China, sometime probably in November, although it may have gone undiagnosed before that, November to December. And then it spread from China to, the spread, by the way, here, anytime you see the purple circle is China. Anytime you see a line coming out of China, that's a known spread from China to someplace else. So an individual here who was sick with SARS-CoV-2 had a virus that they know from sequence analysis came from China. Now, we're able to keep tracking these viruses over time, where they go and where they came from, because viruses don't stay the same throughout time. They mutate. And it seems like SARS-CoV-2 right now is mutating at about 1 to 2 bases per month. So we can literally track these changes from one sick person to the next and follow the trail of the infection. And so the first sick individuals went out of China into Europe, the United States, Australia, and Southeast Asia. There was then a very large amplification of cases in Europe, and a number of individuals then traveled from Europe to the United States, bringing SARS-CoV-2 with them. At that point in time, the SARS-CoV-2 infections in the United States were well underway and really blooming. And currently, the United States has the most COVID-19 cases in the world and is also the biggest exporter of COVID-19 in the world. We send it to more areas of the country as we travel there than any other country does at this point. We simply have more cases. As of this morning, when I looked up the numbers, there were over 2.6 million individuals infected worldwide, over 181,000 deaths, and in the United States, we had over 852,000 individuals infected and over 43,000 deaths. It's certainly more than that, as we'll see in a little bit. We can have individuals who are not yet expressing symptoms and are infected. So there are certainly a lot of individuals out there who are infected who we don't know about yet because they've not either gone to the hospital or aren't showing very many symptoms. All right, so obviously this virus has spread pretty significantly since its origins in bats. And so the next thing I want to talk about is how we slow the spread of SARS-CoV-2. This is also absolutely in the realm of fact, not fiction. The best way to slow the spread of SARS-CoV-2 is to keep yourselves from infecting other people. And so first of all, wash your hands. And the best way to do that is with soap and warm water for at least 20 seconds. And for those of you, most of us don't have second hands anymore attached to clocks in the walls. So the best way to time 20 seconds is to sing happy birthday twice. Or for those of you who are SU students and alumni and know the alma mater, you can sing the alma mater chorus once. Dr. Kate Hastings in communications told me that that was a way that you could do that. And I timed it out this morning and it was true. Fortunately for you all, I am not going to subject you to my dulcet tones at this time. So you can just take my word for it. If soap and warm water are not available, using hand sanitizers is a perfectly acceptable alternative. You need to make sure that they have at least 60% alcohol. But again, remember that soap and water work. You don't need to stockpile these hand sanitizers. As we said, stay at least 6 feet apart from other individuals when you are out in public. More is better. And if you're in an area with stay at home alerts or orders, please do that. The best way to stay away from people is really to stay away from people. When you're out in public, do wear a mask. The CDC, and I've shown the connection or the HTML link here, but if you just go to the CDC's coronavirus page and look up face coverings, the CDC has lots of ways that you can make your own face cloth. Fortunately for me, most of them don't involve sewing. I don't know how to sew worth a darn. Some of them just use t-shirts and rubber bands. They're pretty simple. It's a great way to find out how to use it. The reason that you need to wear a mask is because of those water droplets. When you wear a mask, the water droplets that you are exhaling mostly stay inside that mask instead of infecting other people. So again, this is a way to keep other people from getting infected and in turn, slowing the spread and minimizing the chance that you'll get infected. And then of course, you can use disinfectants. You should use disinfectants to disinfect your environment, those things that you're touching regularly, particularly if you have a sick person or have been around sick people. All right. So let's move on to the current status of testing. Ultimately, to stop the spread of COVID-19, we're going to need to be able to have accurate and plentiful tests available so that we know who is infected, and we'll need to be able to treat it effectively and immunize effectively in order to prevent it long term. Currently, the only way that's approved by the FDA, the Food and Drug Administration, to test for the presence of the virus is through nucleic acid amplification tests. So I think the one that everybody's familiar with, the most common one, is shown here in the figure where you've got these nasal swabs. They stick this up inside your nose. If you're infected with the virus, that will pick up some of those viral particles. They then send that off to a lab and they look directly for the presence of the virus RNA. If that RNA is present, that means you are infected. There are also new tests out that are testing for antibodies against the virus. And the FDA has only approved 4 of these and is quite explicit that none of these are approved to test for infection by SARS-CoV-2. And I want to explain a little bit why that is so you understand. When we get sick, when we get a viral infection, it actually takes our immune systems a while to make antibodies against that infection. So the early stages of an infection never involve antibodies. So if somebody goes for testing with an antibody test early on in their infection, they're going to test negative even if they have the infection. The next thing is that there are a couple of different kinds of antibodies that are typically made in response to viral infection. The first is something called IgM. That's made very early in the infection and could be diagnostic in some cases, again, it would be more, I shouldn't say it could be diagnostic, it would be indicative if it was there of a early presence of a viral infection. That type of antibody is not typically associated with long-term immunity. So one of the things that people are talking about with these antibody tests is to determine who might be immune to the virus and who might not be immune to the virus. The other type of antibody that these tests can test for is something called IgG. And this is the type of antibody that tends to be made even later in an infection, so a week to weeks into an infection, and that is produced by cells when somebody does have protective immunity. Now, just because somebody has this kind of antibody doesn't actually mean that they have protective immunity. We simply haven't done the research to know that yet. So currently, the appropriate use, I should also say that the FDA has, like I said, approved 4 of these, but even for those 4, the incidence of false positives is about 5%. So you'll tell somebody that they're infected and have antibodies when they don't about 5% of the time. The rest of those antibody kits that are out there can be as bad as 30% effective and 70% wrong. So most of them really aren't very good at all. And even the ones that are good can only show the presence of antibodies at a particular time. They are useful. I think one of the things that make people most excited is that you can use those kits in conjunction with what you know about somebody's disease state and somebody's recovery state and look for how long antibodies persist after infection. And that can be really useful information and may help us understand vaccinations in the long run. So again, currently the only real way to know if you're infected by the virus is to use those nucleic acid amplification tests. And as we all know, those simply aren't plentiful enough or available enough yet for us to be able to test everybody that we would need to test to know who is and is not infected. The next area that I want to talk about is sort of the current landscape for developing drugs to treat COVID-19. And I know this is a really busy figure. It simply shows the life cycle of the virus again. And so if I can get there's my little arrow. Right at the top again, we can see our cartoon virus binding to this ACE2 receptor, getting inside the cell, making its proteins, making its nucleic acids, and getting out of the cell. And over here, it's causing an immune reaction, an inflammatory reaction. And I'm just showing it here. You can probably all see there's lots of drugs here in red, and it shows many, but not all of the current drugs that are currently in the pipeline to try to block the virus replication or the virus symptoms in some way. Currently what most folks are doing, there are a couple of areas where people are trying to develop new drugs, but right now, mostly what's being done is people are testing drugs that are currently being used for other viral infections or other diseases in order to try to stop the progression of COVID-19. And the reason that these are the first places that folks are starting is because they're already out there. They've already been through clinical trials that show that what their toxicity levels are and that humans can tolerate them and that they may be effective. Currently, there are no FDA-approved treatments of COVID-19. So what I'm going to be talking about today are drugs that are currently in clinical trials with no or few published results. And again, there's no FDA-approved treatment yet. Some of these are being used for compassionate use treatment, and this is something where you can use a drug that's in the pipeline if there are no viable therapies present. So many of these drugs, actually all of the ones that I'm going to talk about today, are currently being used on patients and are currently being used in clinical trials to see if they can be approved. So the first one that I'm going to talk about is remdesivir. And this is getting a lot of press right now. And it's way down here, right in the middle of the cell. I'm going to try and circle it with my pointer a few times. And remdesivir blocks something called the RNA-dependent RNA polymerase. And this is an enzyme that the virus brings with it that is responsible for making copies of the virus genome. We don't have this enzyme in our bodies, so remdesivir can block that virus without actually doing too much harm to us. It's currently one of the therapies used for Ebola and Marburg virus infections. And the reason that it's getting a lot of press right now, there was just a recent study that showed that it was able to slow the progression and reduce the death in non-human primates. So non-human primates were given this drug and they had much better disease progression. There's also some indications in ongoing human trials that it may be helping as well, but those final results aren't there. Now, the non-human primate study is a non-published study, and I'm going to talk about that a little bit later. So we need to take that with a grain of salt. It's hopeful, but it's not published literature yet, and the clinical trials that show whether or not this really is effective and whether it's safe for treatment in COVID-19 aren't out there yet. The next drug that I'm going to talk about is tocilizumab, and this is something that's used in some cancer therapies to dampen down immune responses to the treatment. And it has its effect over here to the upper right, where we see this inflammatory response happening. Specifically, it interferes with a molecule called interleukin-6 that is one of the cytokines that can help cause that cytokine storm and the very bad inflammatory reactions. So this particular drug is being used to treat the symptoms in individuals who have particularly bad respiratory distress. And then the final drug that I'm going to talk about today really illustrates in many ways the due diligence that we need to do in separating fact from fiction, and that's hydroxychloroquine. This is a drug that's used in malaria treatments and also in treatments for individuals with autoimmune diseases like lupus and rheumatoid arthritis. And this is shown, get my little arrow going again, right here in the middle. So what happens is, again, we said that virus needs to enter our cells, and it enters into a little compartment called an endosome. And in order for that virus to get its genetic material out of that endosome, this compartment needs to become more acidic. And so that's a normal part of what happens. This gets acidified or becomes more acidic, and then that virus can be released into the cell, the virus genome. In malaria treatment, malaria in one of its forms does very much the same sort of thing. It goes into an endosome and needs to be acidified to get out. And in malaria treatments, hydroxychloroquine actually seems to keep that acidification from happening. So it actually is a base, the opposite of an acid, and it enters, its basic component enters the endosome and keeps that malaria parasite from being able to get out into the cell. And so it's hoped that hydroxychloroquine might do the same thing. Now, an early and unpublished result of compassionate care showed that individuals treated with hydroxychloroquine seemed to have a shorter course of disease and fewer deaths than individuals who are not treated with hydroxychloroquine. That immediately was taken up by social media, and lots and lots of people started trying to get a hold of this, remembering that it still really is necessary for individuals with lupus and rheumatoid arthritis. This drug, along with remdesivir and tocilizumab and pretty much everything else on this chart, are now in the midst of well-controlled clinical trials. And there have been 2 recent trials that have shown that hydroxychloroquine may actually not be particularly safe. So it is known to be associated with some cardiac arrhythmias, some really dangerous ones. And since COVID-19 is also associated with cardiac symptoms, one of the smaller clinical trials was ended because there were too many incidences of cardiac arrhythmia with hydroxychloroquine. And a recent study, again, this is an unpublished study that was done on over 300 patients from VA hospitals, looked at the outcomes of individuals who had been treated with hydroxychloroquine and hydroxychloroquine with an antibiotic, and showed that there was no difference between the likelihood that somebody would end up on a ventilator for individuals who are treated with hydroxychloroquine versus those who weren't, and that there seemed to be increased fatality in those who were treated only with hydroxychloroquine versus hydroxychloroquine and the antibiotic, or no treatment whatsoever. Again, this is an early unpublished study, and it needs to be verified. There are ongoing clinical trials that are well controlled that will be used to verify the findings in all of these. So we really do need to be careful about sort of jumping to conclusions too early before the science has had its ability to play its way through. So ultimately, how do we prevent COVID-19? And that will be largely by developing a vaccine. So just for those of you who aren't biologists or aren't very familiar with this, what a vaccine does is it takes a part of a virus or a killed virus or an inactivated virus, and they inject that into you. They give you a shot, or sometimes you have the oral or nasal versions of these vaccines, and they trick your immune system into thinking that you've been infected by the virus. Your immune system then responds to that by making cells that are able to make antibodies that in the future, if you actually are infected by the virus, will inactivate the virus. So I want to draw your attention back to these spike glycoproteins here. These were those red molecules on the surface of the first virus that you saw. Now they're shown as blue. Again, this is where the virus binds to our cells, and they are the most common target for these antibodies because the antibodies will bind specifically to those proteins, and if the antibodies are bound, then the virus can't bind to our cells. So those are called neutralizing antibodies, and that's sort of what everybody is trying to do with vaccines. Currently, there are 1 to 2 vaccines in the very early stages of clinical trials. And so I want to go now, this center column looks at a bunch of different kinds of vaccine strategies, and anybody that wants to ask questions about that or later, I'm perfectly happy, but it's a little bit beyond the scope of this talk and the time that we have today. So I want to talk about sort of where we are with vaccine delivery and how long people think this is going to take. So first of all, if we are able to develop a vaccine to this virus, it needs to go through clinical trials. And in this second arrow here, you can see it says phase 1, phase 2, and phase 3. This basically looks at, in sort of ever increasing numbers of individuals, whether the vaccine is safe, if it causes harm. In phase 2, they really look at dosage and how to administer those vaccines. And in phase 3, they move into much larger trials with more individuals to look at whether it works and whether it's safe. So does it actually cause a person to be immune to the SARS-CoV-2 or not? Each of those clinical phases actually takes quite a long time, months really, weeks to months. Once those are completed, those vaccines still need to be licensed or approved by organizations like the FDA in the United States or other organizations worldwide. I suspect, given the severity of this particular viral outbreak, that those kinds of approvals will come along pretty quickly. Once those things are approved, then you need to be able to ramp that up. And then the next line down shows a factory. So you have to make a lot of a lot of vaccines in order to immunize enough people to get what we call herd immunity, to get enough people immune that this virus won't really spread in the world. And that's going to take a while. You then need to get those vaccines to people and administered. It is not uncommon for viruses to which we've had no exposure in the past to need a booster vaccine. So it may be that a vaccine for SARS-CoV-2 would require a couple of vaccines, and those are usually 3 to 4 weeks apart. And then, of course, it'll take us, even after vaccination, at least a few weeks to have protective immunity to that virus. So ultimately, the time frame is unclear as to when this is going to be. It says in the slide here, 6 to 18 months and maybe longer. I think 6 months is wildly too optimistic. We're probably looking at 12 to 18 months or longer. And the next question, of course, after that is going to be, is one vaccine going to be enough, or do we have to make multiple vaccines, that is, yearly vaccines? And I'm going to take you back to our virus here, and this gets to something we don't know about the virus yet, whether this virus is going to behave itself well, and that is not mutate too quickly, and also the nature of the mutations. And so we know that influenza mutates rapidly, and its mutations to its proteins on the outside of its cells that it uses to bind to us accumulate very, fairly rapidly, and rapidly enough that every year we need to make vaccines against those new strains of influenza. On the other side of the spectrum is the measles virus. It also mutates fairly rapidly, but it turns out that the mutations in those binding proteins are not well tolerated by the virus. In fact, it tends to lose its infectivity when mutations are there. We don't know what's going to be the case with SARS-CoV-2, whether we'll need yearly vaccines, whether one in decades like measles is going to be enough, or if it's someplace in between. So that remains to be seen. Again, we're looking at months minimum before this is happening, many months. So at the bottom of the slide here, the best way we have of preventing and slowing the spread of the virus is through our own behavior. That is, again, those things that we talked about early, staying apart, covering your face with masks, listening to those healthcare professionals that are really experts in the field. And that brings me to really my last part of my formal talk of separating fact from fiction, is that I'm trying to give you some of your own resources for separating fact from fiction for yourselves. So the first 4 bullets there are the organizations in the United States and worldwide that are really responsible for studying this virus, for preventing its spread, and for letting us know how to keep safe ourselves. So that includes the Centers for Disease Control, the National Institutes of Health, the World Health Organization, and your state and local health departments. They all have websites that are largely kept up to date with the most current information. So I encourage you to go there. Those individuals have no ulterior motives other than to beat this disease and to keep us all healthy and safe. The Food and Drug Administration is in charge of certifying and licensing antiviral drugs, the SARS-CoV-2 tests, and vaccines. So if you're interested in seeing what's in the pipeline and what stages everything is at, I encourage that you go to their website and just go to their COVID-19 website. It's all in there. The Environmental Protection Agency, I've put this in here because they're the ones that have the lists of the disinfectants that you can use that work on COVID-19. They've got a long list of that and how to tell whether or not your disinfectants at home will work on this virus. They also have very nice descriptions that debunk some of the ideas that are out there that somehow air purifiers in the home will do anything for COVID-19. They don't stop the coronavirus, and drinking water is not contaminated by the coronavirus. It's not spread that way, in the United States, certainly. So again, the Environmental Protection Agency. All right, so again, what I want to encourage you all to do is to move forward in your interactions. And while COVID-19 is still spreading around the world, using science predominantly rather than social media. I've heard a lot of people recently say, oh, well, they're always making changes in the recommendations. I mean, that means they don't know anything. In fact, we need to remember that only 5 months ago, we didn't even know this virus existed. And so when recommendations change, it doesn't mean that we don't know anything. It means that we've just learned something new, and that the health professionals are making recommendations based on the best and the most current science available. And you should expect that to keep changing as we learn more. So keep watching those sources and be safe.
And we want to thank Dr. Tammy Tobin from Susquehanna University for that very interesting and informative presentation on COVID-19. And stay tuned for more episodes of conversations on COVID-19 in the very near future. I'm your host, Chris O'Rourke. Thanks for joining us. We'll see you next time, and stay safe out there.